with Professor Yogita Bansal. She has already taken a session on impurities or the contaminants from the drug synthesis. And as she is actively involved in the synthesis of new molecules, organic molecules having therapeutic potential, so she is well versed of the approaches which can be used for the green synthesis. So in this session, she will be discussing about the various tools which can be used for the synthesis of organic compounds, so with minimum pollution. So over to Dr. Yogita Bansal, please.
Thank you, Dr. Gulshan. I will just share my screen. Is this visible now?
It is visible. Yes, it is visible.
So good afternoon all. Very good afternoon to all the participants. So today my talk is about green chemistry approach, a tool to control environmental contamination. Yesterday we talked about the pollution which is caused by drug synthesis, various drugs which are synthesized by pharmaceutical processes, how they are leading to the contamination in the environment. So today I will talk about the solution to that. That is a very good solution is green chemistry approach. As we all know that chemistry has improved our quality of life. Although chemistry has improved our quality of life and made thousands of products possible, unfortunately, this achievement have come at a price. Our collective human health and global environment is threatened. So our bodies are threatened, our water bodies, aquatic life, as we yesterday studied that how the contaminants are moving into the aquatic environment. So our aquatic bodies, water bodies are contaminated with large number of synthetic industrial chemicals. So I'm giving you a few examples. You might be knowing about the Minamata disaster, which is caused by dumping of mercury compounds in Minamata Bay, Japan, for about 37 years. It is estimated that about 3,000 people suffered various deformities, severe mercury poisoning symptoms, or death from what became known as Minamata disease. This is the some pictures of Cuyahoga River fires, which were repetitively occurring in 1929, 1949. After some time, this Cuyahoga River fires are occurring because of the effluent from the industrial waste. This is the Bhopal gas disaster. You all are knowing this is the enduring tragedy which occurred in Bhopal in 1984. This is about the Buddha Nala. Buddha Nala, how the name, this is the stinking stream of Ludhiana. Buddha Nala was initially known as Buddha Darya. Now how this name Buddha Nala came into, that over the years, the name got changed to Buddha Nala owing to the sewage, industrial domestic wastes. Owing to the sewage and the industrial domestic waste that is dumped into it. So this these all contaminants, as they are moving into the water bodies, these need to be rectified by some approach, and the green chemistry approach is the best approach which is used. What is now green chemistry approach? It reduces the pollution at its source by minimizing or eliminating the hazards of chemical feedstocks, reagents, solvents, and products. The goal of green chemistry is to create better and safer chemicals while choosing the safest and most efficient ways to synthesize them and to reduce the waste. Now, as these are the various steps which are involved in the chemical synthesis, or that is starting from the raw material, then chemical synthesis, then end products, and then end of the life management. These all steps are these are accompanied by potential risks to the workers, to the suppliers, to the shippers. For example, chemical synthesis is having the potential risk to the local environment, to the global environment. So what green chemistry is doing, it is reducing the risks all along the life cycle of this chemical production. This green chemists, what these green chemists do, they work to ensure that a product will pose minimal threats to the human health or the environment during production, and so that these chemicals can be recycled or disposed. A green chemistry approach is one of the continual improvement, discovery, and innovation that will bring us ever closer to the processes and products that are safe within natural ecosystems. Ultimately, a product should be safely degrade as the biological nutrients, and it should be safely recycled. Now, as these are the various goals of green chemistry, so in 1998, there were two chemists, that was Paul Anastas and John Warner, who proposed 12 principles of green chemistry. They proposed these 12 principles of green chemistry, which demonstrated that how this chemical production could respect the human health and environment while also being efficient and profitable. Now, these are the 12 principles of green chemistry. Starting the very first principle, prevent waste. The waste should be minimized in the environment, in the chemical process. Maximize atom economy, less hazardous chemical synthesis, safer chemical products, safer solvents and reaction conditions, increased energy efficiency, use of renewable feedstocks. One should avoid the formation of chemical derivatives or usage of chemical derivatives, catalysis, degradable chemicals and products, real-time pollution prevention, and minimize potential for the accidents. These are the 12 principles which were given by these two chemists, Paul Anastas and John Warner. Paul Anastas is known as, called as father of green chemistry because he was the pioneer in this field. He gave the term green chemistry. And these are the 12 principles. And now in this following presentation, I will discuss these 12 principles in detail, that how they are, with examples, we will discuss all these principles, that how they are contributing towards the greener environment. So as these all are the 12 principles, I had here shown that these 12 principles are either reducing the human and environmental hazards, or they are improving the resources, use of the resources, or they are increasing the energy efficiency. Now, the yellow colored principles, they are mainly involved in the reduction of hazards to both human life and environment. Whereas the sixth principle, that is designed for the energy efficiency, is for the increase in the energy efficiency, and others are for the improved usage of the resources. These are, I'm giving you some example about the of green chemistry. That is, very basic examples are disinfection of water by chlorination. Initially, water is disinfected. Till now, in the swimming pools, water is disinfected with the chlorine. Whereas chlorine, because chlorine is having an oxidizing effect, it oxidizes the pathogens and kill them. But at the same time, chlorine forms the chlorinated compounds, which are very harmful. So a remedy to this is the use of some other oxidants, for example, supercritical water oxidation in the presence of ozone. Another example where I am giving is of the green processes is the safer dry cleaning method. Very initially in dry cleaning, conventionally dry cleaning was carried out with the usage of gasoline and kerosenes. Then it was overtaken by the usage of PERC, that is perchloroethylene, which is nowadays also used for the dry cleaning purposes. But this PERC is a suspected carcinogen and a groundwater contaminant. So it should be largely replaced by the new processes. New processes which can be used for the safer dry cleaning are usage of liquid carbon dioxide. Usage of liquid carbon dioxide. Now, this liquid carbon dioxide is non-flammable, non-toxic, and a renewable substance. So this is a greener method for which should replace the older conventional methods. This is the another example of the production of beta amylase. The beta amylase enzyme, which is commonly found in sweet potatoes. This is isolated when isolated with the conventional method. This is the comparison of two processes, conventional process and a greener process. In the conventional process, the starting material which is to be used for the isolation of this enzyme was around 6,000 pounds of sweet potatoes, and it utilizes about 1900 gallons of acetone for the extraction. And the significant use of electricity was also required in this process. But when this process is replaced by a greener process, by some other method, this will utilize not only reduce the amount of the starting material, that is, you can see that the amount which was initially required was 6,000 pounds, it reduced to about 2,000 pounds. And in this extraction method, in this procedure, no solvent is required for extraction, and minimal usage of electricity is also required in this process. So this is a greener method and a better approach for the extraction of this beta amylase enzyme from sweet potatoes. See, these are some of the examples of the green methods. So now, as I discussed, that I will discuss all the principles of green chemistry, starting from the very first, and I will give you some of the case studies and examples of all these principles. So we will now first discuss the first principle, that is prevention of waste. It is always better to prevent the waste than to treat or clean up the waste after it has been created. Suppose if in the chemical reaction, the two reactants are involved in the formation of the product, majority of the times that chemical reaction is also involving the formation of some byproduct, or which can be considered as a waste. So one should find the greener method so that the waste produced is minimal. This is the picture which I am showing. Is this picture is of Suahago River again, in which the industrial waste is being dumped into this river, and because of which this river was causing the fires. Now, what is the measure of this waste? How we can check the amount of the waste formed? The magnitude by which the waste formed in the reaction can be measured is the E factor. It is the actual amount of the waste formed in the process, which includes the solvent losses, acid and base used up in the workup, process aids, and in principle, waste from the energy production. So when we compare this waste, that is E factor is amount of the waste in kilograms divided by amount of the product formed in kilograms. E factor is calculated, it is the ratio of amount of the waste formed upon amount of the product formed. So when we compare the waste generated by the different industrial segments, it was observed that the waste generated in the oil refinery was minimal, whereas the waste generated in the pharmaceuticals you see is maximum. As I had discussed yesterday also, that the pharmaceutical industry, it is because it is producing lot of waste, because it is into the synthesis of the molecules, complex molecules, which are used for the humans, which is required for the human use. So these complex molecules have a great demand of quality standards, and these are mainly synthesized by the batch processing methods rather than the continuous methods. And the regulatory constraints are very high, and these products should be of high value. So the waste generated in these, particularly these kind of products, pharmaceutical products or drugs, is comparatively higher in comparison to the basic and the fine chemicals, or in comparison to the oil refinery. So this waste generation should be decreased. Either there are many methods to decrease this waste generation. It can be decreased by either decreasing the number of the synthetic steps or by implementing the solvent recovery systems. Now, how this waste get generated? Why this waste get generated? This waste can be generated by the usage of stoichiometric amounts of acids and bases. Acids and bases are generally required for the nitration, simple aromatic nitration reactions. When benzene is nitrated to nitrobenzene, generally it is carried out in the presence of nitric acid and sulfuric acid. So these acids and bases, they are act as the potential sources of wastes. Many acid-promoted rearrangements are also involved in the chemical synthesis or in the drug synthesis. For example, Beckmann rearrangement reaction or aldol condensation reaction. This we had discussed yesterday also. I have given you the example of aldol condensation because aldol condensation requires aldol condensation. I have given that it is applied in the synthesis of Taxol. Taxol is one of the drug anti-neoplastic agent. So as the aldol condensation is involved in the synthesis of Taxol, aldol condensation is involving the usage of base, that is sodium hydroxide. So this base can act as a potential source of the waste generated in this reaction. Okay, another is Lewis acids, which are used in the Friedel-Crafts acylation reaction. That also we studied in the synthesis of ibuprofen. I explained that very first reaction of the step of the synthesis of ibuprofen involves the Friedel-Crafts acylation reaction. Now, because this Friedel-Crafts acylation reaction involves the usage of Lewis acids, that is aluminium chloride, zinc chloride, boron trifluoride, or hydrofluoric acid. These all may act as the sources of the waste. Various oxidants and reductants can also act as the sources of waste. For example, chromium compounds are generated when the oxidizing agent sodium chromate is used, or when potassium permanganate or manganese dioxide. All these lithium compounds will be generated from lithium hydride, as I given the example of sodium borohydride also, and I discussed the toxicities of boron also. When boron is act as an effluent, or when it moves to the environment, what kind of toxic effect it is having on the human body or on the different microorganisms when it is when it is poured into the sea, how it is having the dangerous effects on the aquatic life. So these all oxidants and reductants, they are also acting as the potential source of the waste. Various halogenation and substitution reactions and solvent losses. Solvent losses is also a waste, that is they are air emissions or aqueous effluents. Now, as I discussed, that waste is the factor, a measure of waste is E factor. E factor was amount of the waste formed upon amount of the product formed. In the case of E factor, we are discussing only the amount of the waste. We are not discussing the quality of the waste. But as the mass, because in the case of E factor, only the mass of the waste generated is taken into consideration. Only the mass of generated is consideration. What is important in the environmental impact of this waste is not just the amount, it is the quality of the waste. What is the quality, nature of the waste must also be considered. So, for example, here as I had shown in the, for example, if 1 kilogram of sodium chloride is dumped into the environment, it is not equivalent to 1 kilogram of chromium salts. Okay? Hence, the new factor, that is environmental impact or environmental quotient, it came into picture, which is calculated by multiplying the E factor, that is amount of the waste formed, into the Q value. Q value is the unfriendliness of that waste. For example, in the case of sodium chloride, the Q value is 1, whereas in the case of chromium salts, the Q value may be up to 1000 because of the toxicity of the chromium salts in comparison to the sodium chloride. Okay, this is because of the toxicity. Not only the toxicity, ease of recycling and many other factors should also be considered while considering the Q factor for a particular waste. For example, ease of recycling, usage of that waste. Sometimes the waste generated in a particular reaction can be used somewhere else also. Okay, for example, hydrogen bromide एक example ले रही हूँ. जिसमें hydrogen bromide is having a lower Q quotient when compared to hydrogen chloride. Because why it is having a lower Q value? Because hydrogen bromide is having the capability of recycling via oxidation to bromine. Because its oxidation to bromine is easier, so it can be recycled. Okay, in another cases, the waste product may have some economic value. For example, in the synthesis of caprolactam. Caprolactam is a starting material for the synthesis of nylons and plastics. Okay, so in the synthesis of caprolactam, ammonium sulfate is generated as a waste. You can say it is generated as a byproduct, but its Q value is low. Why its Q value is low? Because this ammonium sulfate has a capability to be used as a fertilizer. So because it is having some economic value, its Q value decreases. Okay. So then waste, the unfriendliness or the environmental impact of a particular waste will not only be calculated by the amount of the waste produced, but will also be calculated by the nature of the waste, capacity of the waste to be recycled, or by the economic viability of that waste. Okay. So this is, I am giving you one example of a greener process in the synthesis of the allyl alcohol. So allyl alcohol, I had shown here, there are two methods, traditional route by which allyl alcohol was initially synthesized, and one is the greener route. Now in the traditional route, we see that allyl alcohol is synthesized by the alkaline hydrolysis of allyl chloride. Now in the alkaline hydrolysis of allyl chloride, the allyl alcohol is generated along with the production of hydrochloric acid. Hydrochloric acid is a byproduct in this reaction. Okay, so to avoid this chlorinated byproduct, another greener method, alternate method, which was suggested by green chemists was the two-step use, the synthesis of this allyl alcohol by two-step method. Initially, the intermediate is synthesized by utilizing propylene and acetic acid. Okay, then this intermediate is hydrolyzed in the presence of water and converted into the allyl alcohol. In this method, acetic acid is formed as an additional byproduct along with the allyl alcohol. Now, this acetic acid, which is obtained in the second step, can be used in the first step. So it is no waste is formed. Effectively, no waste is formed in this particular two-step reaction or two-step synthesis of allyl alcohol. Okay, so the added benefit I had written is that the acetic acid produced in the second reaction can be recovered and used again for the first reaction, leaving no unwanted waste material. Clear? So this is an example, a very good example, and a simple example where we can show to how we had minimized the waste production. This is one case study which I had encountered, that is sildenafil citrate production. Now, what is this sildenafil citrate? Sildenafil citrate is known as Viagra. It is a selective inhibitor of phosphodiesterase 5. This new drug immediately became a major seller when it came into the market during its very first year of the launch. Okay. Now with rapid sales, and production is to be increased. The pharmaceutical production of sildenafil citrate is to be increased. So a better methods should be generated so that its environmental performance is also good. There should be a better methods. Okay. The conventionally sildenafil citrate, the methods which are used conventionally for the synthesis of sildenafil was it was 11-step synthetic procedure, which gave only 4.2% overall yield. The intermediates which were used, or the reagents which were used in the reaction, was tin chloride, which is a heavy metal, and it is a major environmental polluter. It also required the thionyl chloride as a solvent, which is again having a very high environmental impact. The another compound organic molecule which was used is hydrogen peroxide, which causes burns upon skin contact and is a fire and transportation hazard, especially when come in contact with the other organic materials. Now, and the waste material produced was about 1300 liter per kg of the product. So the E factor in this case is 1300. This is a very high E factor. So with time, the processes are improved so that the waste is minimized. this is how I had shown that how with time the processes are improved so that the waste is minimized. So initially in 1990, the process which was utilized for the synthesis of sildenafil, the E factor was 1300. When 1994, the E factor was reduced to 100, then it was reduced to 22, then it is reduced to 7 liters. Now in the future, they are further targeting to reduce this E factor of the waste generation to around 4 liters. Okay. So the alternative, now what are the alternative methods which are used were, this was the 3-step method and the yield is increased from 4% to about 97%. Reduction in the ratio of the solvent waste per kg product over 17 years from 1990 to this 2020, the reduction of the solvent usage, we see that it is decreased from 1300 liter to 7 liter. Okay. It is minimized by either adopting some different methods which are not using the solvent in that amount, or we are increasing the methods of solvent recovery, or solvent selection is improved. Okay, these are the various strategies which were followed to improve the solvent efficiency. Now, only the solvents like tertiary butanol, ethyl acetate, 2-butanol, and trace of the toluenes are still required to be disposed of by these newer methods. This is the amount of the organic waste, amount of the aqueous waste, and amount of the vapor phase, which is to be targeted. Amount of the organic waste is reducing with the time with these newer methods. Okay. So the target is to green chemistry, that is prevention of waste. Now we are moving on to the second principle of green chemistry, that is atom economy. Now, what is this atom economy? Atom economy is the incorporation of all the atoms of the reactant into the product. If all the atoms of the reactant are incorporated into the product, mean that particular chemical reaction is having 100% atom economy. So the synthetic methods should be designed to maximize the incorporation of all the material used in the process into the final product. So percentage atom economy can be calculated by dividing the molecular weight of the product over the molecular weight of the reactant. So these I have shown the various examples of the reactions. 0% atom economy means no reaction has taken place or something wrong had taken place. Now, in the case of substitution and elimination reaction, or in the case of chiral resolutions by using stoichiometric reagents, we can increase the atom economy. By using use of the catalyst, we can increase the atom economy. Some of the reactions like addition reactions, rearrangement reactions, or isomerization reactions, where all the atoms of the reactants are converted into the atoms of the product, they are having the 100% economy and atom economy, and I have given you the examples of such reactions. The first example is allyl phenyl ether. It is rearranged into ortho-allyl phenol, where all the atoms are present in the product also, of the reactant are present in the product also. So this reaction is having 100% atom economy. Similarly, another is the addition reaction. It is a diol-Zelder addition reaction, cycloaddition reaction, where we are cyclizing butadiene and ethene to cyclohexene. All the atoms again are utilized in the formation of the product, thus having 100% atom economy. Another is the example of the addition reaction. The two reactants are adding to form the two different products, again with the 100% atom economy. So the chemical reactions should be improved so that the improved atom economy is there. Yeah, there I am giving you some of the examples. For example, in the first case, I am discussing the reduction of the ketonic group to an alcoholic group. The carbonyl group is reduced to OH in the presence of sodium borohydride. When sodium borohydride was used as a reducing agent for the carbonyl group, the boric acid is formed and sodium hydroxide is formed as the waste material or as the byproducts. So the atom economy gets decreased because the required product is having only 81% of the atoms, whereas 19% of the atoms get wasted in the formation of the byproducts. If this particular reduction reaction, instead of being carried in the presence of sodium hydride, is carried out with the help of the catalyst, metallic catalyst, that is palladium. Yeah, then this reaction will have the 100% atom economy. Okay? In the second, first one is the reduction example of the reduction reaction. Next one is the example of the oxidation reaction. When the OH group is oxidized to a ketonic group in the presence of the Jones reagent. Jones reagent is the acidic chromium trioxide. When this is reduced in the presence of Jones reagent, the atom economy is only 44%, and the E factor is also greater than 3. E factor is related to the production of the waste, because in this reaction, the chromium containing waste is generated. So the E factor is also greater than 3, and atom economy is very, very less, that is 44%. If this particular reaction is oxidized in the presence of air, in the presence of some catalyst, for example, palladium-2 complex is used as a catalyst in this oxidation reaction, then the atom economy can be increased up to 91% by minimizing the E factor to less than 0.1. And water is the waste which is generated. Okay, so this is a very good alternative for the oxidation reactions to be carried out to minimize the waste formation and to maximize the atom economy. Okay. Another example, another case study which I am taking, which is having improved atom economy, is the synthesis of ibuprofen. So it is this method which is utilized for the synthesis of ibuprofen is having 6 stoichiometric steps, and it is having only 40% of the atom utilization. The atoms which are present in ibuprofen required product is only 40% of the atoms from which we had started the chemical reaction. So this is the alternative catalytic synthesis of the ibuprofen, in which only 3 catalytic steps are used. First of all, the steps are reduced and the atom economy is increased to 80%. Okay, so this is the GIAN method where the atom economy is increased to 80%. So this was about the second principle of green chemistry, that is increase in the atom economy, to increase the atom economy. Okay, now we move on to the less hazardous chemical synthesis. Now, where less hazardous chemical synthesis, wherever practicable, synthetic methods should be designed to use and generate the substances that possess little or no toxicity to the people or the environment. Now, we all know that chlorine has been widely used in the textiles and paper industry, and it is essential in the in many plastics and chemical industries, that is in the formation of PVC. Now this chlorine is produced industrially by utilizing, initially it was produced by utilizing the mercury cell. That this is the chloroalkali process, which was used for the synthesis for the synthesis of chlorine, is utilizing the mercury cell. It was based on the mercury cell. So now what is the disadvantage is that the contamination of mercury waste. We already know that contamination of mercury waste has tragic health and environmental effects. Okay, and the heavy metal accumulation is that Minamata disease. We already, as I had shown the initial in the very beginning of this presentation, I had shown one picture of the Minamata disease, which is caused because of the dumping of the mercury in the river. Okay, so this mercury-based chloroalkali process acts as the source of the mercury contamination. Okay, so this should be replaced by a greener method. The greener method, which is adopted for the synthesis of chlorine, is membrane-based cell. Membrane-based cell is a preferred choice for the chloroalkali process. Here, a membrane, ionic membrane is there in the cell, which is ionizing the brine which is added. Brine is a sodium chloride solution, and it ionizes sodium ions and chlorine ions then combine to give the chlorine gas. Yeah. So this is a better method for the synthesis of chlorine. So this is the less hazardous synthesis of chlorine. The one case study which we will discuss is about the synthesis of adipic acid. Now, what is adipic acid? Adipic acid is hexane dioic acid. As it is shown here, it is hexane dioic acid. Here it is hexane dioic acid. Now, this is one of the most produced commodity or the chemical worldwide. Now, what is the use of this adipic acid is? It is used in the synthesis of nylon 6,6 and very commonly used in the synthesis of a polyamide, that is nylon 6,6. Okay, so it is very commonly used in the chemical, pharmaceutical, and food industry. The conventional method, which was utilized for the synthesis of this adipic acid, utilizes the benzene. This is the conventional method, which I am showing you in this slide. In this, we are starting the reaction from benzene. So in the previous one, we are starting the reaction from benzene. Here it is from benzene, and it is involving the oxidation with an excess of nitric acid. Nitric acid now, because nitric acid is involved in this synthesis, the nitrous oxide gas is released into the or emitted by this process, and it is released into the environment. And this nitric oxide gas is a greenhouse gas, which mainly accounts for 5 to 8% of the anthropogenic nitric oxide emissions. This is a greenhouse gas, so this gas should be avoided. Some alternate method should be adopted so that no such kind of gases are released into the environment. The alternate method which is adopted for the synthesis of adipic acid is, initially the method utilizes the reactant was benzene. Benzene is a carcinogen. Now in this method, the starting material is glucose. The adipic acid is synthesized from glucose. Glucose and intermediate formed are protocatechuric acid, catechol, muconic acid, and then finally adipic acid. Now in this method, water is used as a solvent, and the temperature and pressure required are also ambient. So it is an energy efficient method, and it does not produce any waste. So this is the case study where we are saying that better methods, safer methods should be used. Another case study is in the case of paper bleaching. Initially, the paper bleaching was done with chlorine dioxide, which is again generates chlorinated pollutants, and these pollutants are exceptionally toxic. The alternative method of paper bleaching is with the help of iron embedded hydrogen peroxide, TAML hydrogen peroxide. This alternative catalytic breakdown of H2O2 provides the oxidative equivalent to chlorine dioxide. And this bleaching can be carried out at lower temperatures, and time requirements are also less in this method. So this is the safer method for paper bleaching. Now, moving on to the fourth principle of green chemistry, that is designing of the safer chemicals. Now, the chemicals which are designed should also be safe, or the chemicals which are used should also be safe. The chemical products designed should have minimum toxicity. Now, the safer chemical, there is one list which is provided by safer chemical ingredient list, which is known as safer chemical ingredient list. This is a list of chemical ingredients arranged by functional use class that the safer choice program has evaluated and determined to be safer than the traditional chemical ingredients. Okay, adding the chemicals into this safer chemical ingredient list encourages the innovations and growth in the safer product, increases the market for the manufacturers, and help to protect the people and the environment. Okay. Now, what this safer chemical ingredient list, this gives the color code to the chemical. The 4 color codes which are provided by this list to the chemical are green circle, half green circle, yellow circle, and gray square. Okay. Green circle is the low hazard chemicals. Green half circle is the expected to be low hazard based on the experimental and the model data. Yellow triangle meets safer choice criteria for its functional ingredient class, but may raise some hazardous profile issues. And gray squares are not acceptable. The chemicals which are included in the gray square are not acceptable for the use in the product. So they are not acceptable. Okay. So how these chemicals can be enlisted into this list of safer chemical ingredients is these are the various steps which are to be followed for a chemical to be included in the list of this SCIL. This is the Environmental Protection Agency, which is enlisting the chemicals into this list. Now, when any chemist, it is submitting the chemical or the ingredient, then it is the chemical ingredient is first reviewed for its safety and different criteria for the safety towards the human health and environment. And if it meets all the criteria for the safety, then it is listed into this list. Very recently in 2020, these are few of the ingredients which are enlisted in the say this list SCIL. These chemicals were submitted by American Cleaning Institute. These are the chemicals, surfactants. These are mainly surfactants, octadecanoic acid, alcohols, fatty acids. These are mainly surfactants which are used in cleaning industry. And this American Cleaning Institute, it submitted the list of these chemicals to be added into SCIL. And this EPA, they approved this 8 cleaning product ingredients and added into this list in 2020. So another case study about the usage of the safer chemicals is the, for example, DDT. As we all know that the usage of DDT, the DDT which is used as an insecticide, agricultural pesticide, and as a malarial control agent. DDT is highly carcinogenic and is a threat to wildlife, especially birds, and it has almost led to the extinction of bald eagle production. This is the threat, it is a threat, a maximum threat to the environment. So how this DDT can be replaced by some alternative like spinosad. Spinosad is produced by the bacteria, that is Saccharopolyspora spinosa. This is a pesticide. Spinosad is a pesticide which is isolated or produced from a bacteria. This bacteria is isolated from a Caribbean soil samples, sugar mill. Okay? This spinosad selectively targets the nervous system of the insects, and it is very less toxic to the mammals. It is having, because of its high selectivity, it is having low toxicity to the mammals and thus has a good environmental profile. If we look into the toxicity profile of this pesticide, that is spinosad, in the case of rats and ducks, it is having very high lethal dose. Matlab, at the dose of more than 5,000 milligrams per kg, it is not lethal, even at the concentrations of 5,000 milligrams. Whereas in the case of fish, it is lethal at 30 milligrams per liter. And in the case of bee, where it is required, it is lethal at very low dose, means it is highly selective towards the nervous system of the insects. So this was about the 4 principles of green chemistry. Next, I move on to the very important principle of the green chemistry, usage of safer solvents and auxiliaries. This you might have been studying in all these days, the usage of safer solvents and how the disposal of these solvents, they are posing the threat to the environment. So the safer solvents or no solvents wherever possible should be used. Okay? Why? These solvents, because these solvents accounts for the vast majority of mass wasted in synthesis and processes. Moreover, many conventional solvents are toxic, flammable, and corrosive. Solvents' volatility and solubility have contributed to air, water, and land pollution and have also increased the risks of the workers' exposures and have led to the serious accidents. Recovery and reuse, when possible, is often associated with energy-intensive distillation and sometimes cross-contamination. So in an effort to address all these shortcomings, chemists have started to search for the safer methods. Now, safer methods, first of all, I will discuss that in the pharmaceutical industry or in any chemical process, what is the role of these solvents? These solvents or the water contributes almost about more than 80% to the process mass intensity. Suppose if 1 kilogram of API, by this time you all must be knowing that what is API. API is active pharmaceutical ingredient. Suppose if we want to synthesize 1 kilogram of API, 100 kilogram of material is required, in among which solvents and or the water contributes to the major amount. Okay, so there is the emphasis to do the research and to reduce the use and hazards of these solvents. Now, why these solvents are required in the manufacturing or synthesis of these chemicals is they are required for various processes like mixing, for selectivity, to improve the reaction rates, for the scalability of a particular reaction, isolation and cleaning. Okay, and the greener alternatives is that no solvent, either one should avoid the usage of solvent, that is no solvent is used in the particular reaction, or the solvent should be environmentally benign and safe, that is water if possible. One should use maximum usage of water should be done as a solvent, or the solvent should be easily separable and safe. So supercritical fluids is one of the solvents which are in this category, or the solvent should have zero volatility, and ionic liquids are the solvents which have zero volatility. Now, there is a guide. A guide was My Green Lab. This resource guide was provided by the collaboration of Beyond Benign, Millipore Sigma, My Green Lab, and a network of chemistry faculty. They created a guide with the data resources for practicing greener chemistry in laboratory studies. A guide was created by all these three, that is Beyond Benign, My Green Lab, and Millipore Sigma. All these three generated a guide which facilitated the practicing, which gave us the practices which should be used for the greener methods. Now, who is this Beyond Benign? Beyond Benign is a non-profit organization dedicated to green chemistry education. Beyond Benign disseminates green chemistry concepts and best practices globally to science educators, scientists, and the public at large through their education programs that are designed. Okay? And what is now My Green Lab? My Green Lab is again fundamentally and permanently improving for the sustainability of scientific research. This is again a non-profit organization which is for the sustainability of scientific research. Now, this Millipore Sigma is a leader in the life science. They are solving the toughest problems by collaborating with the global scientific community. These are dedicated these are more dedicated to make the research and biotech production simple, faster, and safer. So all this, the guide also provides the green chemistry principles and lab practices. These principles and lab practices are mainly provided to carry out the experiments for the undergraduate organic chemistry labs. Now, what information do this green chemistry guide provide? In this green chemistry guide, Pfizer created an internal solvent selection tool. This solvent selection tool categorizes the solvent based on their worker safety, process safety, environmental, and regulatory considerations. Solvents are categorized into preferred, usable, and undesirable solvents, and are also presented in the solvent replacement tables. The solvent replacement tables are also presented. Okay? So they are categorized into preferred, usable, and undesirable process. Through the internal use of these solvents, suppose undesirable solvent is a pentane and the alternate provided by the Pfizer is heptane, or if suppose in the case of dioxin, the alternate prevented is 2-methyl tetrahydrofuran. By the usage of these alternatives, solvent selection tools, 50% reduction in the chlorinated solvent was seen, and 97% reduction of the undesirable ether usage was observed. And the usage of heptane by replacing pentane is also observed observed in many cases, okay, with the help of this Pfizer selection tool. This is the another tool which is provided by GlaxoSmithKline. It is a solvent selection guide which is developed a solvent selection guide for the use by the medicinal chemists. Now, according to this solvent selection guide, these solvents are again selected on the basis of waste, environmental impact, health hazards, flammability, explosion hazards, reactivity, stability, and their life cycle score. They are categorized into solvents having few issues, solvents having some issues, and solvents having major issues. So they are also providing a selection guide, GlaxoSmithKline. Now, this is the Beyond Benign, which has created, again, Beyond Benign has created a greener solvent. These all tools are provided in this greener solvent guide. Now they are again categorizing the solvents into hazardous, problematic, and the preferred ones. Okay, so the solvents which are colored red are hazardous, which are colored with yellow color are problematic, and the greener solvents are the preferred solvents for the organic synthesis. Now, green chromatographic solvent selection guide. This is the Amgen developed a guide in which DCM is सबसे नीचे देखो तो black color में DCM is the solvent, in which DCM is replaced with the greener alternatives. Now, why DCM is replaced? DCM is known to be associated with respiratory and cardiovascular toxicity in humans, carcinogenicity, and genotoxicity. So this DCM is replaced with the other solvents, and their elution profile is compared with the DCM in methanol. The guide is, it is comparing the eluting power of the different greener solvent mixtures with reference to the DCM and methanol. So this is also provided in this guide. So this is the elution power of some basic compounds and acidic compounds in comparison to the DCM and methanol with some greener solvents. So the solvents can be of wider utility. They can be required for scalability. They can be required for greeniness. It is up to us that how to select a solvent. So the solvents which are in the green region, which are having all these three properties, wide utility, scalability, and greeniness, are the best solvents which are to be used in the processes, chemical processes or the pharmaceutical processes. So this was about the usage of the solvents. So next we move on to the sixth principle of green chemistry, that is increase in the energy efficiency. Now, the chemical processes should be designed so that the energy requirements is minimized. So that energy requirements are minimized, and that is the synthetic methods which are to be carried out, should be carried out at the ambient temperatures or the pressures. Mainly energy is required in the pharmaceutical processes or in the chemical industry. The energy is required for heating, cooling, separations, and pumping. So the reactions should be designed in such a way that they are carried out at ambient conditions. Ambient conditions mean at room temperature and at atmospheric pressure, so that the energy usage is minimized. A very basic example, which I am going to discuss here, is that the hot plate, which we are using in our laboratories, which is a hot plate heating an oil bath at about 110 degrees C. It uses about 100 watts of power, which amounts to about 72 kilowatt per hour in 1 month. And a vacuum pump uses 3 times more energy than this hot plate. So by minimizing the use of these equipments in the laboratory, how we can minimize that? We should turn off the equipment when it is not required. It will also help to conserve the energy and to save the money. Okay, in our academic labs, the amount of energy, and आपको लगेगा कि ये थोड़ी देर के लिए switch off कर देंगे तो कितना फर्क पड़ेगा, ठीक है? In the academic lab, maybe वो उतना ज़्यादा फर्क नहीं पड़ेगा. But when we move on to the large scale, okay, the money savings are multiplied and the energy efficiency is multiplied. So it matters a lot. Things become bigger. So make a habit to conserve the energy by switching off the equipments whenever they are not required. Okay? So this is one of the case study which I am taking here to give you, to explain you how the energy minimizing is very effective. Atorvastatin, you might have heard. Atorvastatin is a cholesterol lowering drug which suffers from energy demanding synthesis as a result of two cryogenic reactions. In this synthesis of atorvastatin, two cryogenic reactions are required. These reactions are to be carried out at -70 degrees C. ठीक है. Now, so -70 degree C के लिए बहुत ज़्यादा एनर्जी चाहिए उतना टेम्परेचर मेंटेन करने के लिए. So new biocatalytic synthesis is developed which uses the enzyme data and shortens the process by removing two energy intensive chemical process. So by this, energy is minimized. So what are the other methods to minimize the energy? As I had discussed, that mainly the processes requires the heating. Heating is the main thing which is required. Okay? So the alternative methods of heating can be used to save the energy. What are the alternative methods? Is sonochemistry, microwave chemistry, or photo-assisted. The reactions, sound waves can be used to promote the chemical reactions. Okay? This is mainly used in the production of triglycerides from methyl transesterifications. Microwaves can be used. Microwaves are very commonly nowadays used in our laboratory setups for the synthesis, as they are promoting, they are decreasing the reaction time to very large amounts. Okay? They are avoiding the unnecessarily prolonged residence times. Okay, and photo-assisted reactions can also be carried out. For example, sun can be used as a catalyst. The photo-driven acylation for the production of valuable synthetic intermediates and commercial fragrances in bulk. This is one of the application. It is also photo-assisted reactions are used by BASF to develop automotive primary coatings and a precursor, which is a precursor readily able to cross-link with photo irradiation as opposed to its conventional energy-intensive, thermally driven variation. So these are the various methods which can act as the alternative to the conventional heating sources. Microwave is a very good factor which we are using in our labs also. So what are the advantages? So I am basically discussing the microwave reactors in organic synthesis, applications of microwave reactors, how these are advantageous in the organic synthesis. They are enhancing the rate of reaction, they are increasing the yields, greater reproducibility, and improved purity, and reactions can be carried out in the no solvent conditions also. The reaction times are shortened and lower temperatures are required. Okay? Now, these are some of the examples of very basic reactions: hydrolysis, oxidation, esterification, decarboxylation. Hydrolysis, for example, the very first, benzyl chloride when hydrolyzed to benzyl alcohol. If it is hydrolyzed by a conventional method, by utilizing some heating source, suppose hot plate, then this hydrolysis will require 35 minutes. And when the same reaction is carried out in the microwave, this reaction can be achieved in 3 minutes only. Similarly, the hydrolysis of benzamide to benzoic acid by conventional route, it utilizes 1 hour, whereas in the microwave conditions, it utilizes only 7 minutes. So this is a very energy efficient method. Okay. Oxidation, example of oxidation is given: toluene when oxidized to benzoic acid. When oxidized to benzoic acid, refluxing is required for around 10 to 12 hours. तो क्या कितना कुछ हो गया जब 10 से 12 घंटे के लिए आपकी रिएक्शन लगी हुई है. देखो, मैन पावर इज रिक्वायर्ड, यू हैव टू मॉनिटर द रिएक्शन, देखना पड़ेगा, छोड़ के नहीं जा सकते हैं. ठीक है. And energy is consumed for 10 to 12 hours. But if the same reaction is carried out in the microwave conditions, this reaction can be achieved in only 5 minutes, just 5 minutes. So it is saving both the manpower and energy of the human being as well as the energy of the resources. So next is the example of the esterification reaction, which can again be carried out in just 6 minutes, and another is the decarboxylation, which again in the presence of the microwave can be carried out in very short period of time, that is just 12 minutes, and the yield is also increased. In some of the cases, you will observe that the yield of the product is also increased. So these are some of the reactions, examples of the reactions which are carried out in neat environment. Neat means no solvent is required. Sometimes for a particular reaction, a solvent is required, but when that reaction is carried out in the microwave conditions, no solvent is required. We ourselves are also performing the reactions, and we are doing those reactions in the absence of the solvent. So these are the examples where the reaction is carried out in the presence of neat reactions are carried out, or some reactions are carried out on some solid mineral support. Okay, pre-absorbed into the either an essentially microwave transparent, that is silica, alumina, or clay, or is strongly absorbing inorganic support. So some solid supports are used. So the advantages of these are they are easy to handle, no special equipments are required, high reactivity due to catalysts, reagents on porous supports. They are safe, no flammable solvent is involved, and they are environmentally benign. Okay. Although there are some limitations of these methods also. So I will discuss now one of the case study of Dow Chemical Company. Now, this case study is of interest because Dow Chemical Company is the largest US chemical company, nowadays called Material Science Company. This company is it is into the process of developing newer methods to minimize the energy or to increasing the energy efficiency. It was seen that the consumption of this energy, the largest component of the production cost of this of this company was mainly because of its energy consumption. In its global operations, Dow uses the energy equivalent to 8,50,000 barrels of oils per day. This is the more oil consumption of some countries, that is Netherlands and Australia के केस में वो पूरी country इतनी oil consumption करती हैं जितनी ये एक company ने अपनी oil consumption कर दी for the processes, pharmaceutical processes or chemical processes which they are carrying out. So now with the time, Dow is trying to improve its energy consumption or to reduce its energy intensity. It has reduced its energy intensity by 22% through the structured program targeting process improvements. Now, how this structured program targeting process improvements, what are the initiatives which are to be taken to improve the energy efficiency? These are the various steps. One should improve the existing processes, that is environmental control and optimization, reducing gas flaring, improving the yields of raw materials, commercializing of the new processes. Commercializing of the new processes, for example, they are the commercialization is this. I had shown in the picture a new HPPO plant at a BASF site in Belgium was started in 2009. With this new process, in this plant, they are producing propylene oxide. And this propylene oxide is, in this generation, the wastewater is reduced to about 80%. Okay, and they are reducing the waste generated. So another steps which are to be initiatives which are to be taken is recycling waste. Dow is recently began operating a novel system for reusing the municipal waste, which I had shown the second picture here. Recycling the landfill of gases for energy. Landfill waste is structured. So recycling of the waste is carried out by the Dow. Another steps which are adopted by this company was investing in the renewable raw materials. This company started investing in the renewable raw materials. Dow is making the liquid epoxy resins. Now, what are these liquid epoxy resins? These are used in coatings, including marine and automotive applications. The new process which involves this epoxy resins is making epichlorohydrin from glycerine. It is in the formation of epichlorohydrin. Traditional method, the epichlorohydrin was formed from propylene, and the newer method, the epichlorohydrin is formed from glycerine. Okay, now in this for newer method, in the traditional method, the derivative of oil or natural gas liquid is used. Propylene is obtained from a natural gas liquid. It is utilizing the biomass. Whereas in the newer method, glycerine is used. Glycerine is a byproduct of biodiesel production. So it is an alternative fuel which is made from a renewable vegetable feedstock. So by using this new EPI process, Dow will now reduce the wastewater generation from the production facility by more than 70% as compared to the conventional technology. Okay. So the last, another method which is used by Dow is creating products that enable energy savings. the different examples of these processes are construction, automotive, packaging. All these are the various products. So this was the case study of dough. Next, I move on to the seventh principle of our green chemistry, that is use of renewable feedstock. One of the example I had already discussed in the previous case of dough, the renewable feedstock in which glycerine is used in the case of instead of propylene for the synthesis of epichloride. Now, what is these renewable? The raw material or feedstock should be renewable rather than depleting. Many chemical processes are using biomaterials for the synthesis of highly functionalized molecules, mainly biomaterials, carbohydrates, proteins, lipids. They are converted into petroleum products. Petroleum products are then converted into olefinic products, and then they are converted into the highly required, highly functionalized molecule. It may be a chemical, it may be a pharmaceutical drug. So these they are mainly utilizing the petroleum products, which is mainly utilizing the biomass. So the newer methods should be used so that the biomass is reduced. As the biomass production, suppose in nature, is 180 billion metric tons per year, and only 4% of this biomass is utilized by the humans. And the other 96% of the biomass is utilized as the building blocks for the chemicals. They are also used in the formation of polymers, adhesives, production of phenolic chemicals, or they are converted into the polymers, lubricants, detergents, which are required in the various types of the industries. So this utilization of the biomass should be reduced and use of the renewable feedstocks should be carried out. This is one of the example which I am taking, that the polymers, synthesis of the polymers from the renewable resources. This is the polymer polyhydroxyalkanoates. They are broadly useful family of natural, environment friendly and high performing bio-based plastics. The development of microorganisms which produce these PHAs are from renewable feedstocks such as cornstarch, cellulose, hydrolysate. The microorganisms have proven to be applicable to the conventional commercial equipment and can even be recycled using this same equipment. So with the help of these microorganisms, this can be synthesized. They can be used in the biodegradable products such as credit cards. They are comparable with polyolefins, which are made from petroleum feedstocks in terms of strength, melting. Now we can replace this petroleum polyolefins or polymers which are obtained from these petroleum feedstocks with some other polymers which are obtained from the renewable resources. This is how, this is again an example where the polymer, because the polymer production is the major source of greenhouse gas emissions also. It is not only utilizing the feedstocks, it is also a source of the greenhouse gas emissions. So newer polymers should be formed so that it should shift towards the renewable carbon feedstocks such as biomass and carbon dioxide. Here I am showing you the example of synthesis of polycarbonate using phosgene. Here the phosgene is used, and in the greener process, the alternative method is utilization of diphenyl carbonate in the synthesis. Now the another example which I am showing is the polyether carbonates. These polyether carbonate polyols, they are synthesized from carbon dioxide. They are synthesized on the industrial scale by utilizing carbon dioxide. Now, when we are using carbon dioxide, the impact of global warming will also be reduced because this is a greenhouse gas. Carbon dioxide is utilized in the large amounts. Utilization of carbon dioxide as an alternative to carbon feedstock for polyols is motivated for the potential to reduce the greenhouse gas. Thus, it is reducing the fossil fuel depletion as well as greenhouse gas emission. Okay. Another is polyurethane. These polymers which are synthesized by these methods, they are having the application in various industries like footwear production, paint industry, biomedical applications. These are the various industries where these polymers are used. This is the another example which I am giving for the synthesis of tetrahydrofuran. In the synthesis of tetrahydrofuran, acetylene and formaldehyde is used. So this acetylene and formaldehyde, instead of acetylene and formaldehyde, it can also be synthesized from a biomass. which is having sugars, and there is tetrahydro. This is an alternative method. Now, this was about the usage of renewable feedstocks. Now, coming to the eighth principle of green chemistry. Now, eighth principle of green chemistry is to avoid the usage of chemical derivatives. Avoid the usage of blocking or the protecting groups for the chemical reaction. Now, why these protecting groups are required? They are required to carry out some selective reactions. If suppose our starting material is having two sites of reactions, and when we are carrying out the reaction can be carried out, or that particular substitution or addition can be carried out at both these two sites. Both the two sites are susceptible to that reaction. So an undesired product will be formed when both the sites are reacting. Okay, but we require only one side to react. We require the selectivity. Then what we will do? We will protect one site with the help of some protecting group. Okay, and then we will carry out the reaction. We will block one site which is not required, and then we will carry out the reaction and to obtain the desired product. But for the desired product, first protection is required and then deprotection is required. So additional 2 steps are added to carry out such kind of reactions. Okay, so there we should develop newer methods where the usage of these chemical derivatives should be avoided, as they are increasing the steps for the chemical reactions. This is one example in which penicillin G is utilized for the synthesis of some semi-synthetic penicillin. Now, in this penicillin G, the usage of chemical derivatives was conversion of penicillin G to 1, then conversion of that 1 to 6 APA, then conversion of this 6 APA to some semi-synthetic penicillin of therapeutic utility. Initially, this penicillin G is first protected as its silyl ester, and then it is reacted with phosphorus pentachloride at -40°C to form a chloroimmediate, that is intermediate 1, which on subsequent hydrolysis gives the desired 6 APA. Okay. This synthesis is largely nowadays replaced by a newer enzymatic process, which is utilizing the third reagent. Third reagent is the enzymatic process. It is using penacillase. The synthesis occurs in water at just above the room temperature. The new synthesis has many advantages over the green perspective. From a green perspective, one of which is that the silyl protecting group is not required. More than 10,000 metric tons of 6 APA is made every year, and much of it by the greener enzymatic process. So this is a fantastic example of green chemistry making a real difference to the environment. So we are moving on to the 9th principle of green chemistry. This is the usage of the catalysts. Catalysts, now what are these catalysts? Catalysts are effective in small amounts and can carry out a single reaction many times. They are preferable to stoichiometric reagents which are used in excess or carry out a reaction. Catalyst ka role, so I think everybody might be knowing that catalyst is used to reduce the activation energy of a particular reaction. It makes the reaction faster. Conversion of substrate into a product is carried out by utilizing lower amount of the energy. Okay, so this is the pictorial representation of utilization of a catalyst. Here the green, when it holds the reactant and the product, and it provides a stage for reactant to convert into a product. Yeah, and then it leaves. So catalyst is consumed initially in the reaction, and then it is reproduced in the reaction. Okay, so this I am giving you one example again of this I had discussed previous also, that reduction of carbonyl compound into the hydroxide in the presence of sodium borohydride. When we are using sodium borohydride for the reduction, boron and sodium hydroxide are produced as the wastes. Whereas when this reaction is carried out in the presence of catalyst, the amount of waste can be reduced. Okay. So this is the another example of where the catalyst usage of catalysts is reducing the is reducing the or is increasing the atom economy. Here specifically, I had shown that how they are increasing the atom economy. These examples of catalysts I had discussed in the atom economy also. The reduction with metals, metal hydride reagents, oxidation with permanganates, manganese dioxides, chromium reagents, and wide variety of reactions like sulfonations, nitrations, Friedel-Crafts acylations, employing stoichiometric amounts of mineral acids and Lewis acids. All these are atom efficient catalytic processes. All these are atom efficient catalytic processes. While these catalytic reactions, I will discuss, this is again a classical aromatic chemistry and non-classical aromatic chemistry, which is using the catalyst. But one have to take care that while these catalytic reagents appear to play an impactful role in the development of the greener processes, there are always a couple points on the flip side of the coin to consider. For example, or for instance, a reaction employing a catalyst may not be necessarily green, since the greenness of the catalyst itself should be considered as well. The catalyst itself used should also be green. Okay, so one should consider that also, that it is not, is it environmentally benign? Is this itself a toxic? One should consider these things also. Then, moreover, lifetime of the catalyst also matters. A catalyst, in theory, perform a reaction an infinite number of times, but in practice, it loses its effectiveness after a certain period of time. Okay, so while considering the catalyst, these things should also be taken care of. So moving on to the 10th principle of green chemistry, this is into the designing of the chemicals and products to degrade the after use. Now, degradation is very, very important. Once the chemicals are done providing their main function, what we will do, we will end up, that chemical will end up into the landfills or wastewater treatment plants where they can enter the water, soils, and airs of our environment. Okay, or from where they can be taken by the animals or the humans. The biggest challenge is making that chemical that are stable during the usage, but don't persist in the environment. That chemical should, when used, should be stable, but when disposed, should be degradable. Okay, the chemicals should be degradable. Another important thing which is to be considered is that we want the breakdown product also to be non-toxic and non-persistent. So it is important to remember that there is a different reason a chemical reaction can break down. It can be due to the reaction, photodegradation can occur, hydrolysis can occur, various degradation processes can take place. A common example which I had taken is of the cutlery, that is single-use plastic. We all use this cutlery, and what we do is when we use cutlery, we use it for 10 minutes and then we throw it, toss our fork into the trash, or it sits into, and then it sits into the landfill for about 1,000 years. Microplastics leach into the water and end up in our body, potentially hazardous to us, as well as the terrestrial and the aquatic ecosystem. The plastic waste, a ton of money is also wasted. In 2010 alone, manufacturers used 191 million petroleum barrels to make this plastic goods. So we can now have an idea that how this plastic cutlery is imposing an environmental threat. So instead of this plastic cutlery, a bio-based cutlery should be used, which is made from stems which can be made from the stems of corns or sugar canes, plant oils, animal fats, straws, wood chips. Generally, nowadays we observe in the parties that wooden spoons are used or wooden knives are used. Okay, so these natural materials, they are converted into PLA, which is a bioplastic. Now, this PLA, which is a bioplastic, traditional, it is better than the traditional plastic, and it is a biodegradable. Another example of this biodegradable I am giving is that a biodegradable, linear chain biodegradable PHBV, that is polyhydroxybutyrate O3-hydroxyvalerate. This is a biodegradable polymer, which is an alternate to polyethylene and polystyrene. Okay, so the linear version is biodegradable, and in the case of this sodium dodecyl benzene sulfonate, PLA, I had given already the compositable cutlery. I had given already an example that PLA cutlery is compositable cutlery. And in the case of detergents, in the case of soaps, this sodium dodecyl benzene sulfonate is a common detergent which is often used. used for this linear alkyl benzene sulfonates. Now, looking at its structure, we can see that linear alkyl chain with a benzene sulfonate attached to it. Okay, it is useful as a detergent because it has a polar head group and a non-polar alkyl, making it a surfactant. Okay, but LAS, that this linear sulfonates, these linear chains are biodegradable, whereas the branch chains are not biodegradable. Why this branch chain is not biodegradable is that in the case of the branch chains, microbes cannot perform, because of the presence of these branches, microbes cannot perform the beta oxidation, since they are no good sites where the reaction can be initiated. Therefore, these branch detergents have to be phased out in most developed countries, and they should be replaced by these linear detergents. Now, moving on to the 11th principle, very interesting principle, real-time analysis. Most of us have driven a car before. Okay, picture yourself driving down the highway in the car that doesn't have any windows. Just imagine you are driving a car which is not having any windows or rear view mirrors. It is very hard to drive such a car, and it will lead to an accident. Okay, so nowadays windows and mirrors are added to a car. Now, what these windows and mirrors they are doing, they are enabling the driver to monitor its surroundings in a real time and allows to react and adjust. Okay, now can we perform a chemical reactions to its desired product without monitoring it? No. Our organic chemist, when he is performing a reaction, he have to monitor the reaction. I always ask my students to monitor every 10 minutes, every 15 minutes, or every half an hour, depending upon the type of the reaction they are performing. So what we need to do, the analytical chemistry here is used for the monitoring of the chemical. A simple example of monitoring of the chemical reaction is utilization of the TLCs. You might have the chemistry people, the chemistry participants who are here and which are involved in the synthesis of the organic reactions. They might have many times used this TLC for monitoring of the reaction. Okay, so these analytical chemistry, it is used for identification, monitoring, measurement, and characterization. The real-time analysis for a chemist is the process of checking the progress of the chemical reactions as it happens. Okay? So the green analytical chemistry methodologies which are used for this real-time analysis are X-ray fluorescence detection for multi-metal matrix, solid phase extractions, supercritical fluid extractions, they can be used. So the replacing batch reactors on the large, medium, and even small scale has distinct advantages. Continuous flow reactors, which are monitored real time. They have precise control of reaction condition. In these continuous flow reactors, one have precise control of reaction conditions, reproducible reaction outcomes, and one can minimize the waste and provide the increased reaction safety. Now, windows and rear, coming to the initial example, windows and rear view mirrors enables the driver to monitor its surroundings in the real time and allows to react and adjust. Similarly, analytical methodologies, which are acting as windows and rear views, enable a chemist to monitor a chemical reaction in real time and allow for the adjustment. If this real-time analysis would have done, now we move on to the 12th principle. If this, here I was just talking about that if this real-time analysis is done, we can prevent many hazards from occurring. Moving on to the 12th principle of green chemistry, which is inherently safer chemistry for accident prevention. One should utilize the inherently safer chemistry for accident prevention. Now, substance and the form of substance used in the chemical processes should be chosen so as to minimize the potential for chemical accidents, including releases, explosions, and fires. Accidents can be avoided by minimizing the hazards. Approaches to design safer chemistry can include the use of solids or low vapor pressure substances in place of volatile liquids. Other approaches include avoiding the use of molecular halogens in large quantities. Continuous flow processes can help to minimize the chemical hazards. This is one of the case study which I am showing, which explains the designing of the safer polymers for the use in the aeroplanes. So in this, the PHA is polyhydroxyamide. Now this polymer can be molded into seats, bins, and wall panels. This is a safer polymer. Example is PHA. It can be molded into seats, bins, and wall panels. Now, this PHA is synthesized under milder conditions. The temperature PHA is synthesized under milder conditions, and it is decomposed into the fire-resistant PBO, that is polybenzoxazole and water upon heating. It gets decomposed into a fire-resistant polyoxazole. So this polymer is very commonly used for the aeroplanes. So because we are not using the safe chemicals, it led to the tragedy in Bhopal, as previously discussed, that in 1984, a tragedy happened in Bhopal, which was an industrial accident in the history. It was the worst industrial accident in the history, in which 40 tons of methyl isocyanate was accidentally released. Now, this methyl isocyanate, because this is a toxic chemical, okay, when this was released, 40 tons of methyl isocyanate was accidentally released when a holding tank, why it was accidentally released? When a holding tank, which was holding this chemical, was overheated at this pesticide plant, Union Carbide pesticide plant, located in the heart of the city of Bhopal. Now, when this methyl isocyanate gets leaked into the environment, this methyl isocyanate was having the toxic effect. This was a very toxic chemical, which was having the effect on the respiratory system, and it is toxic to many organ systems in our body. Okay? It is cardiovascular toxicity, respiratory toxicity, many kind of toxicities are associated with this chemical. So when this chemical was released, it caused 15,000 people, death of the 15,000 people, and hundreds of thousands of more than were injured. Chemistry tries to avoid the things that explodes, light on fire, and are air sensitive, etc. When these things happen in the real world, lives are lost. And this is the very heartfelt example of Bhopal gas tragedy. So another example which I am discussing about inherently safer chemistry is the alkylates, usage of alkylates. They are the mix of high octane, low vapor pressure, branched chain paraffinic hydrocarbons that can be made from crude oil through well-established refinery process and are highly favored as streams for blending into the gasoline. Now, these alkylates, conventional methods which are used for alkylates, but they are typically produced from isobutane and light olefins. So this requires the use of liquid acid catalyzed processes such as hydrofluoric acids. Hydrofluoric acid, we all know, is extremely toxic, and when released, it forms clouds that can be lethal for up to 5 miles. So the AlkyClean technology, what it is doing, the AlkyClean solid acid alkylation process was generated, which produces high quality alkylate without using liquid acid catalysts. The solid acid alkylation process is safer for both people working directly in the production and for the people in the area surrounding the production facility. Because why it is safer? Because it is not using that toxic hydrofluoric acid. There are also environmental and economic benefits since neither acid-soluble oils nor spent acids are produced. So this AlkyClean technology, which was used for the generation of this alkylated product, it is environmental and economic safe. It is both having environmental as well as economic benefits. And for this technology, the LB Marley and in collaboration with CBI won the Green Chemistry Award from this Environmental Protection Agency for their inherently safer alkyl clean process technology. So this is in 2016, they won this award. At the end, I am giving an example of the synthesis of dihydropyrimidone. Dihydropyrimidone is a pharmaceutical compound that displaces medicinal properties as vasodilatory calcium channel blocker. These dihydropyrimidones can be used as anti-anginals, antivirals, antibacterials, and anti-inflammatory agents. More than one synthetic scheme have been proposed for its synthesis. Here I am proposing two synthetic methods, route 1 and route 2. Okay. Compare the two. Now you have, what the question I am giving you is, you have to compare these two synthetic routes and analyze which pathway is greener. And in your comparison, you have to be sure to consider and compare all the aspects of the reaction. So this is a question to all the participants, if listening, that which is the greener route, route 1 or route 2 for the synthesis of dihydropyrimidine? Which is the greener route? Can anybody answer?
Route 2, I suppose.
Very good. Saravjit ji, it's very true. Route 2 is the greener pathway, which should be utilized for the synthesis of dihydropyrimidone, because in this case, the yield is improved. First of all, the yield is improved. The temperature used is almost same, although, but time required for the reaction to carry it out is 15 minutes. So the energy efficiency is highly increased for this method. Moreover, in the first method, ethanol is used as a solvent, whereas in the route 2, no solvent is used.
No solvent is used.
Very right. No solvent is
But instead of that, you are using catalyst.
It is carried out in the presence, instead of the acid, it is carried out in the presence of zinc chloride.
So it is not having any adverse effect on the...
It might be having, so we will have to weigh. We will have to weigh that which is the relatively greener method.
Greener method.
Yes. Okay. So thank you, dear all. Thank you for your patient listening.
I have one question here.
Hanji.
Like some slides that you were talking about the replaceable chemicals.
Replaceable chemicals, yes.
Like in case of benzene, toluene is used. Yeah?
Yes.
There was one table which you were showing there. So my question is like in certain biochemical reactions, these chemicals are used, which you are mentioning as non-safe. Unsafe for use. So can in those biochemical reactions we replace the one which you are suggesting and we are getting the same kind of results? Like in certain enzymatic reactions, also, some chemicals like which you were mentioning there are used.
Yes.
So
these are the very right. It may not be case in all the reactions, but nowadays benzene, if we are talking about benzene, the usage of benzene is obsolete nowadays. We are not, in fact, the chemical reactions which are utilizing the benzene as one of the reactant or as a solvent, they are not promoted. Okay, na? These reactions. So the usage of these kinds of which are highly objectionable, they should not be carried out. Sarvjit ji? Am I able to answer your question?
Yes, ma'am, but I think we need to check on this, that whether we are getting the same kind of results means to that extent or...
As I had mentioned the examples of replaceable solvents, we have given the options, alternatives for the solvents. So there must be the alternatives for the reactants also, what you are saying. Right?
Yes.
Very true. Very true.
Thank you.
Welcome. Thank you.
So if there is any query from any other participant, any feedback, any comment, any suggestion? This has been a great session covering all the aspects of green chemistry, how we can minimize the pollution carried out by the chemical reactions, And of course, we cannot avoid it completely, but yes, we can minimize it. So by following these approaches in routine laboratory experiments at undergraduate level as well as at a postgraduate level or in research, we can sensitize the students to carry this message that we have to follow the green chemistry rules so that we can protect our environment. Thank you so much, Dr. Yogita, for your kind presentation and for your exhaustive presentation. Thank you so much. Our
Thank you, Dr.
next session is with the Professor Yolanda. She will be joining in. Yeah, she is already joined. Let me see if she is available. So Professor Yolanda is the third women speaker of this day. And as in the morning, I said that so in the morning, as I said, that we have three speakers on this Women Day. And last one is the Professor Yolanda. She will be, because we have finished this session around 10 minutes earlier. Professor Yolanda, are you here?
Yes, I'm here. I was listening the previous talk. Do you want that I start?
Yeah, first of all, very happy Women's Day.
Okay, thank you very much. Thank you very much. Well,
you are the most learned, most gorgeous, and most, beautiful women in this forum.
thank you very much. Well, I'm going to share my screen. Let me see. One moment. Have too many doors.
So in this session, Professor Yolanda will be talking about some geographical information system, indication system, through which we can identify quantify the risks and how we can see the extent of contamination and how we can prevent it. So this is going to be, a very new and refreshing session.
Well, everybody can see my screen.
Yes, Professor, it is visible.
Perfect. And
Perfect.
if you want that I start. Well, first, good morning. Well, good afternoon, everybody. Then I'm going to start with this last class of today, and I'm going to talk as Professor Gulsan has kindly mentioned about new things like, for example, risk assessment, the geographical information system to map contamination, and also a little bit about the decision support system. Let me say that this is a new starting point regarding these classes because this time I'm going to go a step ahead, and then as before, you have had many days of course that has explained you everything regarding contamination. I see line of the course that was a very interesting course, and I think that almost every aspect related to contaminants has been covered. How to monitor them, you have a lot of classes mentioning analytical tools, showing you how to perform LC, EC, mass spectrometry, etc. And now in this class, I'm going to start off talking about what to do with this data to really assess and control the environment. Then let me say that this is a very applied class about things that could be done, assessed with our data. Then I think that is a very interesting class because it gives you a clear idea about what we can do with the data that we get in our studies. Then I have divided this session in 3 parts. The first one is about the risk assessment to establish hazards to the environment. Second would be about the geographical information systems. Today I will make just a very short presentation just to illustrate you about the possibilities of the system, because tomorrow we will have a kind of practical class where you will see many examples about things that you can do with a geographical information system to map the contamination. And finally, I will talk slightly about the decision support systems that will help to the environmental managers to take decisions. Then I'm going to start with environmental and ecological risk assessment. And well, maybe first question that appeared is what is the environmental risk assessment? Well, the environmental risk assessment is, let me say, a scientifically understood process for evaluating the ecological risk posed by a particular stressor or contaminant. One important thing about this is that the environmental risk assessment must be systematic and also must be based on scientific data. Then it's a way to combine the concentrations that are found in the environment with the knowledge that is already in place about the potential toxicity of these contaminants for different organisms, ecosystems, etc. Then this risk, as I told you, is assessed through the identification first of the contaminants, the concentrations, flow path, toxicities, or receptors. Then with this we can have not only a knowledge about what are the concentrations in the environment, not only about their spatial or temporal distribution, but about the potential of these concentrations to be danger for the different ecosystems. There are, well, some data that could be different if we talk about environmental risk assessment or about ecological risk assessment. In the case of the environmental risk assessment, you need to know some characteristics of the contaminants that we are working with, like, for example, stability, pollution, seepage, health and safety, closure factors, etc. About this information, I have to tell you that this information must be known, because nowadays to approve or to allow the use of a substances, normally the enterprise that produced them should send to the authorities all this information. Then other aspects of this environmental risk assessment, as for example, stability, health and safety, and closure factors, must be known. The most important problem is about the pollution or seepage, because it depends much more on the characteristics of the environment where these compounds could be produced, storage, or even eliminate. Then in this case, about the pollution seepage is where the ecological risk assessment starts. And in the case of the ecological risk assessment, we have to know the contaminants that reach the environment, the flow path in this environment, and also their environmental fate. In this sense, one question that we can ask to ourselves is how this environmental risk assessment can help us to understand better what happens with the contaminants in the environment. Then we have first to evaluate the design options and to see more or less what are the scenarios where we can find with these contaminants in the environment. And also for this, maybe we need technical support in documentation that are needed in order to approve where it's possible to establish an industry that produces some compounds or that can release to the environment some compounds or not. Then once this has been conceptualized, that means that we know contaminants, possible scenarios, have an idea about the place, etc., becomes the project operations. The program operations involves the design of monitoring programs to ensure that anything that we have assessed could be real in the environment. Then there are also a need of guidance on controls required to meet environmental goals during production, and to assess various management options. Then in the case of closure, there is also a need to design post-closure monitoring programs, guidance for environmental controls during the closure stage, and evaluation of the different closures options. And with this, we can develop industrial activities and activities that could be producers of many environmental contaminants for make an arrangement of all these things. Here I have shown you an example of different environmental risk assessment applications that are established for a kind of mine site. This mine site needs to be closely monitored, and as you could see, there are different things that can be assessed and that can be selected between the better options in order to make these activities safer for the environment. Then you see that in the first step, one of the things that can be done with environmental risk assessment is to establish transport routes, thinking what are the size best roads to transport these products without contaminate the environment. If you look at this photo that I have shown here, you see that there is in this place that is a kind of undetermined place. You see that there are two possible routes, the route 1 and the route 2, and that one of them is much shorter than the other. However, the much shorter route pass just in the side of a lake, and then this could be a problem for the environment if there is any kind of accident with the release of toxic compounds. Then, for example, an appropriate risk assessment will indicate that the route 2 is longer but much safer for the environment. Then environmental risk assessment is something that can help to take this particular decision. In the case of the second photo shows a map about the tailing storage facilities. Then this also can, environmental risk assessment also can help to establish what is the better place for the storage of toxic compounds. Then if you look at the two places that appear in the photo, you could see here that in this place there are not any groundwater, and then instead in the second place there is a groundwater mass that can be contaminated by the leaching of compounds from the storage facilities. Then it can help to establish that this site would be safer than the other if you want to storage several things. And the same happens with the shipping options. For example, if you have bulk samples, it's worse than if you put these samples in containers. Then to make a proper environmental risk assessment about the locations, about the possibility of leaching, about the possibility of runoff, for example, if the place is affected by heavy rains, could be very interesting in order to keep safe the environment. And of course, you have to transport to avoid any problems of release of substances with accidents, etc., or contamination. That is the uses that of environmental risk assessment. The environmental risk assessment has a methodology that must be followed strictly. And the first, you have here a scheme of this methodology with all the aspects that should advance in it. And the first aspect is the problem identification. The second is to identify what is the receptor of this problem. That means in the first case, the problem could be the contamination of a water body. Then what the receptor of the identification would be fish. Fish could be also contaminated by this water and die. Then once we have established the problem and the receptor, it's very interesting to do an exposure assessment. That means to know the amount of compounds at which this fish is exposed. For example, in this case, let me say we can, for example, determine concentrations of contaminants in water, in sediments, and in the own fish, and establish whether these concentrations could produce toxic effects or not. That is the fourth step of this process, the toxicity assessment. And finally, the fifth step is the risk characterization. I don't want to be too long in this class because, as you see, there are many aspects and we have many things to mention, but I would like to follow all these 5 steps and then to show you not only this example, but also a few examples about what we do in our laboratory with risk assessment. Then the first step is the problem identification. That means what can be the endpoint of have a kind of enterprise or kind of facility that can release contaminants to the environment. Then if we look at what is this problem, we have that, for example, in the case of a facility that, for example, in the case of a mine that produce several toxic metals or organic compounds, one of the problems is the possibility that in the tailing storage facilities, some seepages will be produced and then they can reach waters and contaminate it. Other potential problem that these facilities can have is the transport of the products. And of course, for example, if this transport requires to have in their own facility a full storage. All of them can be the cause of the environmental contamination. Then it's important to establish what is the receptor of this contamination. Then, for example, I have put all these examples. First, could be lake ecosystems, like fringing woodlands, the alophytes communities, such lake micro and macro invertebrates. There are also the ephemeral creek ecosystems, like the vegetation communities, the livestock, some rare and endangered flora and fauna that they are everywhere, and also the aquatic communities. And finally, the water resources. And the water resources, in addition to have the problem that they affect to environmental, to aquatic biota, they also have the problem that these water resources could be a source of drinking water for the human being, and in the last term, they can affect the humans, like pastoral stations, residents, the town water supply, also the cattle, and then all of this has a number of problems. Then it's important to make an exposure assessment, and you have here how an exposure, and a very simple example of how exposure could be assessed. There are two ways to assess the exposure. One is to analyze the concentration within the aquatic biota or the human being, etc. And then you know how much amount of this contaminant is inside the human being. Or there are also some methods to determine indirect exposure assessment. That means to establish the concentration in air, groundwater, surface water, soil, everywhere, and then establish what are the most important routes of exposure, and also what are the concentrations that could be estimates in the human being. If we talk about these two types of exposure assessment, the most precise one is the determination of the concentration within the organism of the biota or of the human being, etc. But this is also the most difficult way to see this concentration, because sometimes the techniques that must be used are highly invasive, and in the case of the human being wouldn't be ethical, and in the case of the biota, sometimes they are highly complicated, especially as we enter in the problem of make this analysis in this rare fauna and flora that is critically endangered. Then the indirect exposure assessment is very, very frequent, and you have here an example. Here we have 4 different mediums. Sometimes are considered more, sometimes less. I think that this exposure assessment has an important part that depends on the opinion of the person who is making this risk assessment, and that there are some things that must be carefully considered in order to not make it too much complicated. So I told you in this case, we have 4 mediums that has been considered to make this exposure assessment: air, groundwater, surface water, and soil. You have also in the second column the flow paths of this assessment. And then you have wine, airborne dust, etc. That imagine that this compost could be volatilized at least sporadically in air. And the exposure mechanism in this case would be inhalation. Then in the case of water, it's important to have an idea about the paleocanals that connect one water mass with other, the leaching, the seepage, etc. And then the exposure mechanism could be both ingestion, if this water is used for drinking water, or also thermal contact if is used for showers in swimming pools, etc. Regarding the surface water, almost happened the same. So I think that in part of the world, the drinking water or the water that we drink is surface water. Then also ingestion and dermal contents. And finally, we can have as a medium soil where contaminants could be in soil, established in the soil, pore water absorbed or maybe by capillary rise, and again ingestion and dermal contact and exposure mechanism. Then with this, knowing the concentration in these four media and the percentage that can pass to the human being by one of these exposure mechanisms, we can estimate the exposure of any, well, not only the human being, but also any organ. Then, for example, here we have shown a kind of conceptual model where it's possible to see the different routes of exposure. You see here, for example, the exposure point is the release of some packets with some toxic substance. In the term, we haven't mentioned what is this, and then could be released by volatilization, and then it would be in the air. Their presence in the air will depend on the prevailing of the wind direction. That means that, it can have differences depending on the places. Then also our release mechanism is the release to soil. And of course, other release mechanism, as you could observe, is the leaching to the groundwater. If you look at this scheme, I have selected, it was available on the internet, but I have selected because it's very clear. And you could observe that this part marked in red means the different sources of these contaminants. Then once in soil, one exposure point would be the food chains, because from soil, think that contaminants from soil can pass to the vegetation and can be taken in the different by the biota. Other exposure point is water, because if this compound has released to the groundwater, some concentrations will be in this water. And if this groundwater, as is in the case of this slide, is only used for cattle and for other things that are not drinking water, then in this case, the human could be a receptor, mostly by the inhalation, because the contaminant is in the air. Then this conceptual model tries to show you that this even that there are different degrees of exposure. If we release or expose any toxic substances to the environment in one or other way, all the different all the biota and even the human being is exposed to these toxic compounds. And then there is a need of the next part is the toxicity assessment. That means that it's important to see whether these compounds could produce or not toxic effects on the biota or on the human being. And then it's important to have knowledge about all these things that are in this slide. First, the toxicity. That means that it is needed to know what is the action mechanism of this compound. I think that to make a toxic effect, a substance should arrive to the action place, and this is not always achieved. And then should be taken into account what is the mechanism of toxicity, because it depends of this mechanism of toxicity depends many things and many factors. For example, as the type of organism that can be affected by this toxin. Then other important aspect to consider regarding toxicity assessment is the bioavailability of the compounds. That means whether this compound is easily released in an environment where it's easily uptake by the organism. For example, if we have a compound that could be easily dissolved in water, then this compound, once dissolved in water, could be easily intaken, for example, by fish. In any other way, the fish will have thermal exposure breathing, and also exposure through the food. Then it means that this compound has many possibilities to reach the receptors of this toxicity. Other two important aspects is if the compound could be bioaccumulate and biomagnify. Bioaccumulate means that this concentrate in the body because the compound has a higher affinity for the tissues of this living organism than for the external medium. And other thing that also can happen, and that is the more dangerous one and requires some monitoring, is the biomagnification. The biomagnification means that this compound is so happy in the organism tissues that is getting more and more accumulated through the food chains. That means that the small fish has some concentration, and once the big fish eat the small fish, this concentration is accumulated and biomagnified in the body of the big fish. Then these are factors that must be taken into account for the toxicity assessment, because depending on them, toxicity could be higher or lower. And finally, the last step of this process is the risk characterization. You have here an example of how this risk characterization is performed, and you see different likelihoods that must be taken into account, several consequence criteria that must be into account, and below you have the results of this risk characterization. For example, here we have a descriptor that means the exposure. The exposure could be almost 3 times likely, possibly, unlikely, and rare. And then you could see if the exposure is almost 3 times and is expected to occur in any circumstances, well, in principle, this compound shouldn't be too toxic, because otherwise we'll have several problems. Likely only occurs sometimes, possibly is means that the risk of exposure will be low, unlikely low, and if it's very, very low. Then you have here other consequences that are here taken into account. And well, in the case of the consequences, you see, like if the exposure is quite common, then the consequences commonly are low, because it means that all the organisms are exposed many times. But if we make a mix of these two things, you could see like the consequences of an exposure could become, when the concentrations are very high, extremely dangerous for the different organisms. Then we have characterized the risk that the compounds represent to the environment. I have here some examples of how this risk assessment is performed that has been done in our laboratory. We have some experience making some risk assessment, especially for the aquatic biota. And in this case, these data are from a study where we study the risk quotient. The risk quotient is, let me say that the way to estimate whether the environmental concentration could be toxic or not for the different aquatic organisms. Then some of the legislation mark that it is important to use this risk quotient, that is really a relation between the exposure and the potential toxicity, for 3 trophic levels in the aquatic food chains that are first algae, green algae, exactly, second Daphnias, Daphnia magna, and finally fish. Then commonly this risk quotient is made divided. The concentration, the environmental concentration in water by the toxicity that affect to the organism. Then, as toxicity, one of the problems to calculate the risk quotient is the lack of availability of clear experimental toxicity values. The two most common toxicity values is the lethal concentration 50. But the lethal concentration 50, that is the concentration needed to kill at least the 50% of the organism, is really a value that represents acute toxicity. The common nowadays, the current situation in which the environment is quite well protected means that, well, not so high concentration appears in the environment. In a normal environment that is controlled, the concentration of these of toxic compounds are low. Then sometimes these LC50 values are, let me say, little representative of the real situation in the environment. That means that aquatic biota or any other type of biota in the world is exposed to low concentrations of the compounds, but during a very long term. And other thing that is little take into account in the different environments is that is not only one toxic compound at low concentrations, but many toxic compounds at low concentrations that appear together in one sample. Then this is something that should be taken into account. And if the risk quotients want to be calculated for a real situation, the better parameter to measure the toxicity is the non-observable effect concentrations. That means the lowest concentration, or sorry, the highest concentrations that doesn't produce any toxic effect in the organism along all their life. Well, this is a value that is very difficult to find for different pesticides, because or for different contaminants, because commonly the different points are complicated, and then this is one of the problems that this assessment has, that you never know the origin of the toxicity values that you are using to make this calculation. In this sense, I have to recommend you the United States Environmental Protection Agency has a database that is called pesticide ecotoxicity database, that is able to give you acute and chronic toxicity values of almost all the compounds used of almost all the toxicants based on the estimation, on theoretical estimations. This ecotoxicity database is freely available. I think that the abbreviation is EcoSAR, and you can download it and work a little with these toxicity values if you wish. They use a kind of algorithm to calculate the chronic concentrations, that is the mean value between the non-observable effect level and the concentration that produce a chronic toxicity. And even that these are theoretical values that doesn't has been experimentally calculated, have the advantage that you can calculate them for a very wide range of contaminants in the same way. That sometimes is something that happens, that the results of the environmental toxicity studies are very, very variable depending on the type of experiment, species, etc. In this case, we have applied this risk quotient to the La Albufera Natural Park. The La Albufera Natural Park is a natural wetland that is 10 kilometers below Valencia, and that has a very nice environment, but that is, of course, highly contaminated by the close pressure of the city. And then in this case, we have studied not only algae, daphnia, and fish, but also we have made a study about the different trophic levels, the different ecosystems, and the different organisms that form part of these ecosystems that are in the natural park. Then you have here an example of the results that we obtained. Here you could see that we study especially some pesticides, because in this area, in order to make it sustainable, to preserve the ecological value altogether with the service that they offer to the population, there are a lot of rice fields. And then the main problem of the area are the pesticides. Then here we have a study what happens with these pesticides and the different trophic levels, and you could observe that there are many, many places in danger. I haven't mentioned before because sometimes I talk too rapid and I miss part of the information, but this risk quotient is considered that presence danger when it's higher than 1. When it's between 0.1 and 1 means that, well, these concentrations could be of concern and then that need deep monitoring. And finally, when it's lower than 1 means that, of course, you can do other things. Then you could observe here, like, for example, some insecticides doesn't give a risk quotient for Daphnia magna, but instead they could be very toxic for the insects, etc., etc. Then this shows that this type of risk quotients, risk assessment, are very basic and give results only for very high concentration. Here I have also shown an example. In this case, we have used the 3, instead of making the risk quotient for pesticides, we have made the risk quotient for pharmaceuticals, because also there are a lot of contamination by pharmaceuticals in the area, because it's very close to the city of Valencia, and the city of Valencia has about 1 million people living in the area. And you see, this is the conventional risk assessment. The risk quotient is also called hazard quotient. And well, you see here what happens with many, many pharmaceuticals, and as you could observe, really compounds that appear to be dangerous. This is performed at the average and at the maximum concentration. You could observe that really, in this case, caffeine, and in the case below, where we are working with highest concentrations, caffeine, and also tramadol has given some problems for the biota. But instead, if you make a more wide assessment of the real species that are living in the area, you can find that these compounds could be toxic for one or several of them. And then in part, we'll explain what is happening with these loss of biodiversity in the natural ecosystems that nowadays is considered as so important, because it's one of the things that is very important to preserve to ensure the good ecological status of the environment. Other things that can be done in order to establish the toxicity, because sometimes we don't have any idea about whether this compound is absorbed or not, is the bioaccumulation test. In this case, I would like also to show a little, and with this I will finish with risk assessment and with these examples, because we have also to talk about other aspects. But this is a bioaccumulation test in mussels, in which we work with mixtures of contaminants, just to see this effect of the mixture of many compounds at low concentration. In this case, we try to reproduce a real environmental case, and then we select the Mytilus gaioprovincialis, the mussels that are common of the area, and then these mussels were exposed to 20 contaminants of different origin. If you see, here is a very particular mixture in which we have several pharmaceuticals, like for example, vidagliptin, atenolol, acetaminophen, salicylic acid, etc. We also have several pesticides like chlorphenifos, chlorpyrifos, imazalil, terbutilazine, etc. And we have also several perfluorinated compounds like the perfluorooctane sulfonate. The idea to make this mixture and the selection of these compounds for the mixture was once we have monitored water, soils, sediments, and many other parts of the environment, in order to see what compounds were present and more or less their concentration. In addition to this, in this bioaccumulation test, we make of course a control, but we have two problem groups. The one problem group that was exposed to this mixture of contaminants at a more or less environmental concentration, and other group that was exposed to this mixture of contaminants together with several microplastics and polyethylene. Then we could observe in these experiments, we have first an exposure state in which the mussels were exposed to this mixture of contaminants at a constant concentration during 28 days. And once we have this exposure state, we also preserve these mussels during a depuration state that take place during a total of 58 days. During this depuration state, the water was changed, the water was without any contaminant, and we see what happens with the concentrations in these mussels. Then one of the problems that we have in this study is that we need to develop special analytical methods to analyze the different parts of the mussels. We made the study into matrices coming from these mussels. The visceral mass of the mussel was complicated because it has a very high content in proteins and in lipids, and the hemolymph, that means the water that surrounds the mussel, and this requires to develop specific extraction methods. In the case of the visceral mass, we use a modification of the Kitchers, and in the case of the hemolymph, we use a solid phase extraction method based on the precipitation of several phospholipids. And here I show you the results We get interesting results for almost all the compounds that were in the mix. I don't want to extend too much, and then I just want to show a couple of graphics. In the first one, we have the concentrations of PFD8 perfluorodecanoic acid, a perfluorinated compound, and you could see the red line represents the concentrations of the the red line represents the concentrations, the masses that has been exposed to pharmaceuticals and microplastics, and the blue line represents the masses that has been only exposed to the pharmaceuticals. As you could observe in the case of this PFDA, and also in the case of the other perfluorinated compounds that were tested in this study, looks like there is a difference between the masses exposed to microplastics and or not, because looks like the microplastics helps to accumulate some of these perfluorinated compounds. Instead, in the case of the terbutylazine is one pesticide. If you look at the graphic, look like the microplastics or the masses exposed to compounds and microplastics show a little lower accumulation than in the other case, but instead it affects the depuration phase, and in the depuration phase also the compounds release slower from the mass. These are preliminary studies, and then well, we have some theories to explain this, but really they are not a clear explanation, and we are pending of several additional experiments. Then just to show here the last summary of this result, you have the different constants that were calculated in this study, the distribution constant, the absorption constant, and the bioaccumulation factor. And also during the depuration stage, we were able to calculate some half-life of the compounds. And then you could observe that some of these compounds, like for example, PFDA, PFOA, and PFOS, clearly show lower concentrations and lower half-life in the masses exposed only to the mix of contaminants without microplastics, and instead the concentrations in the presence of microplastics and also the half-life enlarged quite. This could be due, or we suppose that is because these compounds can be absorbed to the plastics, and then they, in this way, change their characteristic kinetic. And also you could observe like there are some compounds as chlorpyrifos and terbutylazine that can be bioconcentrate in the muscles, and that increase very much its life. In this case, this compound gives higher values when there are no microplastics in the environment. Then this at least shows that the relationship between the microplastics and the other contaminants is really complex in the environment, and that they are able to absorb many compounds and to give many problems. Then this is just what happens with again terbutylazine regarding the concentrations and PFPA, and you could see as this type of behavior has been repeated almost everywhere. In this case, we are talking about the hemolymph of the muscles. You see that there would be a sign respond to this behavior that bioconcentration factors are always higher within the group not exposed to microplastic, and instead, in the case of the perfluoropentaenoic acid, the result is completely reversed. That means that the accumulation factors are higher in the case of exposed muscles. With this, I finish with this part about risk assessment, and I'm going to start with the geographical information systems. Regarding the geographical information systems, I'm only go to show you very small part to show what is a geographical information system and who is interested in this system, just because we will devote tomorrow 2 hours to this system. Then I don't want to make it too. Well, one thing that is very clear is that nowadays we have in 2 different worlds. One natural world that we are trying to preserve, and that if the human being wouldn't be in the earth, would be self-regulating. And one constructed world that is whatever we have created. We have created so much, and in a so important way that we have coined the word Anthropocene, Anthropocene, to define as a new geological era where the human being has impacted all the world that is more or less managed. And these 2 worlds are increasingly in conflict, and then we have the problems to represent geographical systems. What is the context? Well, that we need to have 2 perspectives over the world. One is a global perspective, and to see that I see this world as a whole, then we can establish pattern linkages, threads. For example, I will put you just an example to make this clearer. Remember that I explained what happens with microplastics in the environment, and that I show a map of the world because all the plastic that finish in the oceans are accumulated in some places just because the oceans are interconnected, and these places depend slightly on the different currents that move in the ocean. Instead, this is very interesting possibility. We can look at the world as a whole to establish all these things, pattern, linkages, trends, etc. And instead, sometimes, and because of other interests, we are interested just in managing several places, or small places, or places restricted to a very concrete geographical area, as for example, watersheds, communities, neighborhoods, etc. Here I have shown you an example of one of these management that could be of interest and that can give information. For example, here is something, and in addition is something that has little to see with the environmental problems. This is just to open your mind about the possibilities of the geographical information system. This is a representation in the world of the seropositive pregnant women at the Kenyan surveillance places. And then you could see well, how this look like. You have a temporal evolution because you have the pattern of the different places in different years. This is a quite old example, but it's very clear. And you could observe there has been an evolution in these different places that the seropositive pregnant women has increased much more in just 10 years, and that this increase has started by the places with a high population density. If you look carefully at these maps, you have the information about the percentage of these seropositive women according to one survey carried out in these places, and then you have also a layer that shows the population density in the different areas. If we observe this temporal trend, we can see that highly populated areas are a place of increase of the seropositive pregnant women, and that this increase along the years have passed from the highly dense populated areas to the others, but in any case, is always higher in these dense populations areas. You can also here bar graphics that shows you the percentage of these women in each one of the different big cities. Then you have here a good and nice picture about the information that these geographical information systems can offer and can combine and their utility. These geographical information systems has also the possibility of abstracting the real world. What I try to mean with this? Well, you have here what happens, and then if you look at that city, the city has parks, the city has water points, the city has streets, different land uses, etc. Then you can separate each one of these aspects in one different layer in a way that then you can combine all the layers that you need. For example, if you just want to see the different districts, you can use one of these layers and not the other one. This is very helpful when, for example, you want to eliminate several layers just to see more clearly the possible interconnections, for example, between the different water sources, or you can add it if you want to see any relationship with them. This is something that these systems allows. After this presentation of the results, maybe you are asking what is a geographical information system? Well, it's just a computer-based system that includes software, hardware, people, and geographical information. Then one of these GIS can create, edit, carry, analyze, and display map information on the computer. That is the strong possibility of this, because it has many additional possibilities to perform spatial analysis that I will explain tomorrow. Well, what is important this? Because involves geography. Keep in mind that 80% of the government data collect is associated with some location in the space, and then this allows to introduce this information in this space. Then the information attributes or the characteristics data that can be used to symbolize and provide further insight into a given location, and the system that involves a seamless operation linked to the information or to the geography. Then this is interesting because sometimes you can see interesting movements, interesting trends in transport and fate, etc., etc. This requires hardware, network, software, data, and operational procedures. Then a geographical information system is not just a software. Currently, they allow too much things. And it's not just for making maps, they allow other important functions. Who use these geographical information systems? Almost everybody, the international organizations, the private industry, the government, the non-profit organizations, and the academic and research institutions. You have here these interesting parts, and then you could see different organisms, especially. This just gives an idea, and but by sure that if you look at least at the international organizations, you could see that it covers the needs of international organizations devoted to many things that are very different between them. Then that is important of the change. And just to finish this class, I'm going to show you a little about the environmental information systems. Commonly, there are two types of environmental information systems. The environmental management information systems that allows you to collect and manage the environmental information. I have to tell you that this GIS environment is one of these environmental management information systems. And also there are some environmental decision support systems that helps to the manager to take decisions. The environmental management information systems commonly storage and retrieval information such as records, data that are needed to support the environmental management system, such as those prescribed by the ISO 14001. This is very important because these environmental management information systems are relatively new in the market, and one of the problems that they have is that they have to manage many informations and many big data series, and then are quite difficult to manage. And one of the problems that they have is that there is not an universal system. You will see that many. I will show tomorrow with the geographical information system that many of the programs has to be paid, and then they have a cost, and they are not so easily accessible, and then they give several problems. Commonly, these systems are based on a relational database, although spatial databases such as the GIS are beginning to appear in this context. And commonly, well, one of the problems is that they have a need to be integrated. And this is difficult because many of them are not compatible each other. The information that you introduce in one not always can be used in another. And then there are some object-oriented approach that has data, software tools, and a management process, but they give still a lot of problems and are very difficult to manage altogether. Normally, other of the problems is the separation of entities. That means that commonly they are clear. They have some data. These data are introduced in a software application, and then this software application is able to help to establish processes, etc. But the interrelations are sometimes a little complicated. You have here a small example that shows, for example, if you are studying a contaminant, you have data about the employees of the industry. Then this could be managed by a personal database application, and could help in, for example, establish a plan for hazard communication. This could be related with the facility data, like, for example, let me say, if you are producing one toxic gas, then you can include some gas sensor applications to monitor the emissions. But finally, you also need to establish some materials, and you have to purchase a database for these applications. Regarding the environmental decision support systems, there are information systems that helps humans to make environmental management decisions. They are focused on specific decision problems and decision makers, incorporates such active information management operations as modeling, simulations, expert systems, optimizations operate with data, and engage many times spatial data management directly by using geographical information management as the framework. You see, like GIS, decision support systems, etc., are really very interconnected. The main problem with this decision support system is that they are highly complicated, and that historically environmental management data tool has been discrete and disconnected. Then this has the effect of prevent actual decision makers from using them. And if we want good decision, we need to make this data and this tool accessible, because sometimes, well, you will see that GIS could be very easy or could be very complicated. And then when you start at this point that you need to make a special course of a specialization to manage them, then these tools become little accessible for the decision makers, because the use of this tool is not the first priority. Then I have here an example. This example is a decision support system that I will show you about what happens with some spills. And they made a kind of this decision that needs first to integrate several tools, like the Latin hypercube samples, the mathematical process modeling, the linear optimization, expert system, GIS system. Then the second part is to monitor the well neighbor designer, and finally to analyze the low-level waste facility. You can see here, in this case, they are trying to establish a kind of storage facility, and then this storage facility has spills and has several water bodies. And you see these points represents where the contamination has been formed. And you see here, like they can get this score about the cumulative distribution of these contaminants in the area, and then to have this score can help very much to decide if what will happen with the concentration of these compounds in the environment. This has been just an idea of showing you several possibilities of these systems. The system are complexes, but let me say, is the future that comes. And then this needs to be managed, and of course, maybe they need to be more freely available and more used by everybody, not only for the people who is expert in the system. Then GIS is a clearly established system that has some possibilities that I will show you in the next class. In the next class, I will put you several examples, because it must be a kind of programs, and I will show you how to manage with several programs, and also will give you a list of free available programs. And then I hope that this introductory class has been useful. And with this, I will finish my presentation for today. Many thanks for your attention.
Thank you very much, Dr. Yolanda. Thank you very much. It was an you see, very informative session where the use of experimental techniques, systematically designed research, and use of the geographical indicators or geographical information system, how we can create it and how it can be used for control of contamination in a particular area. That's true. We have to think globally but act locally. So this is how we can control the contamination. So if there is any query or any suggestion or any feedback from any participant, please feel free to ask. Anyone? Dr. Gaurav, Dr. Sumit, Gurpreet, Pramila, Seema, Dr. Kuldeep, Simrat.
No, I think that no. They are already tired. Okay.
Okay, so Dr. Yolanda, thank you so much.
Thank you to you. And okay, see you tomorrow.
Yeah, see you tomorrow. We will be having another final 2-hour session on problem solving session with examples on this same topic, risk assessment, introducing problem info, introducing just GIS, and thereafter we'll be having an assessment section session for all the participants. They will be given questions through the Google form, and they have to answer the questions, and based upon their performance they will be given certificates of this course. So this is you see
So how many questions will be there in the paper?
there will be around 60 questions.
60 questions, and the time allotted?
Time will be 1 hour. Not 1 hour, it will be 45 minutes.
60 questions, 45 minutes.
Yes. It's a multiple choice.
Okay, and it will be like starting at 4 o'clock, I think.
Yeah, we will start at 4 o'clock. I will share the Google form before we will share the link for Google form before well before it, but it will be activated only at 4 o'clock.
Okay.
Right?
I think the time provided is very less.
Okay, I will see to it. We will do something so that you get sufficient time. Let's see. I will try to make it 1 minute per question. Right?
Yes, sir. That will be
Either
fine.
we
Thank
will
you.
increase the time or we'll decrease the questions.
Thank you so much.
Definitely. And so make sure all participants, they remain logged in throughout the session tomorrow. And because without this, those who are your friends, just convey this thing to them also. So thank you so much for all of you. If you have any further query, you are free to ask. Okay.
Sir, one request. Can you share the presentations?
Okay.
Particular
I don't have all the presentations PDF,
if
so
possible.
I don't have the PDFs of all the presentations. So whatever I have, I will try to share. And I have to ask it the local coordinator also whether I am allowed to do it or not. Right? But I will try to facilitate to the maximum extent.
Yeah, at least some material is required now, because it is like difficult to grab so many things in short duration of time. It will be helpful for us actually.
And definitely. I will try to facilitate that.
Thank you.
Right. So thank you so much, all of you. Have a great evening and see you tomorrow at 10 o'clock.
Okay.
So tomorrow, actually, we will be having a session on, again, water contamination from a different perspectives. And we'll be having a session on the electronic waste. What kind of electronic waste is being created, and how it can be mitigated, and what are their effects. And then the final session of this course by Professor Yolanda on this problems related to this GIS. And then
We will be having 3 sessions tomorrow also.
3 sessions tomorrow, and then assessment session, final. Right? Thereafter, we'll be having a valedictory. Okay. Okay, thank you so much. Bye-bye.